This mummy was alive for 3,000 years? DNA report says something impossible. This mummy is 3,000 years old. It should be completely dead, completely silent, completely finished. But when scientists tested its DNA, they didn't find decay. They found something still active. Not metaphorically. Not symbolically. Biologically. Cells that should have collapsed thousands of years ago were still responding under the right conditions. That shouldn't be possible. Because after death, DNA breaks down. Cells lose structure. Everything stops. But in this case, something didn't stop. The discovery did not begin as a mystery. It began as routine. A team of archaeologists working in a dry desert basin uncovered a sealed burial chamber, buried beneath layers of compacted earth and stone. The region was already known for its preservation conditions: low humidity, stable temperatures, minimal microbial activity. In other words, a place where bodies can survive time in ways that seem unnatural. But even in such environments, there are limits. Skin dries. Tissue collapses. DNA fragments. The past becomes quiet. Inside the chamber, there was only one body. No signs of disturbance. No evidence of intrusion. The wrappings were intact, layered with care, suggesting this individual was not ordinary. The position of the body was deliberate, almost ceremonial, as if whoever placed it there believed this person mattered beyond death. At first, the team catalogued the find like any other. Photographs. Measurements. Environmental readings. Nothing unusual. Until they opened the inner layer. The tissue beneath the wrappings did not behave like ancient remains. It was preserved, yes, but not in the brittle, fragile way typical of mummification. There was elasticity. Resistance. Even color variation that suggested something deeper than simple drying. One of the researchers reportedly paused, noticing that the skin did not crack under minimal pressure. It responded. That was the first moment things stopped making sense. The body was transported to a controlled laboratory for further analysis. Standard procedures followed. Carbon dating confirmed the age, approximately 3,000 years. There was no error. This individual had lived and died in a time when early civilizations were still defining their identities. And yet, what lay on the examination table did not behave like something that belonged entirely to the past. The first layer of explanation was simple. Exceptional preservation. There are known cases where environmental conditions slow decomposition to an extreme degree. Cold climates, salt-rich soils, oxygen-deprived spaces. These can halt decay processes in ways that appear almost unnatural. Perhaps this was one of those rare cases. A perfect combination of factors that froze biological time. But that explanation began to fracture when genetic testing began. DNA extraction from ancient remains is difficult. Over time, strands break apart, becoming fragmented and incomplete. What scientists usually recover are damaged sequences, useful, but far from whole. In this case, however, something unusual appeared. Portions of the DNA were not just intact, they were structured in a way that suggested stability far beyond what should exist after millennia. At first, the team assumed contamination. Modern DNA can sometimes infiltrate samples, creating false readings. So they repeated the tests. New samples. Sterile conditions. Independent verification. The results did not change. Then came the observation that shifted everything. Under specific laboratory conditions. Controlled temperature, chemical stimulation, certain cellular components within the tissue began to react. Not in a way that implied life as we understand it. There was no heartbeat. No metabolism. But there was response. Molecular activity. Signals that suggested parts of the biological system were not entirely inert. This is where the known explanation began to fail. Because preservation can slow decay, but it does not maintain responsiveness. A preserved body is static. It does not react. It does not change. It does not behave as if something inside it is waiting. So what exactly were they looking at? The deeper investigation moved into a second layer, one that involved not just archaeology, but molecular biology. Scientists began comparing the DNA sequences with modern human populations. The goal was simple: identify lineage, origin, connection. Place this individual somewhere within the known human story. But the results introduced another contradiction. Certain genetic markers did not align cleanly with any known population group. There were similarities, of course, this was still human DNA. But there were anomalies. Variations that appeared rarely, or not at all, in existing genetic databases. It was as if this individual belonged to a lineage that had either disappeared, or had never been fully documented. Now the mystery was no longer just about preservation. It was about identity. One theory suggested that this was an example of an unknown preservation technique, something lost to history. Perhaps ancient practitioners had developed a method of halting biological processes without destroying cellular integrity. A kind of primitive cryostasis, achieved not through cold, but through chemistry. If true, it would mean that ancient civilizations understood the body in ways we are only beginning to rediscover. Another theory focused on environmental factors. The burial chamber itself may have created conditions so precise that decomposition was not just slowed, but fundamentally altered. Unique mineral compositions, atmospheric isolation, microbial absence, together forming a natural system that preserved not just structure, but potential. But neither theory could fully explain the DNA stability or the cellular response. A third, more controversial idea began to circulate quietly among researchers. What if the anomaly was not in the environment, but in the body itself? What if this individual possessed a genetic variation that made their biological systems behave differently after death? A mutation that allowed cellular structures to resist collapse. Not immortality, but resistance to finality. This idea was difficult to prove. And even harder to accept. Then came the breakthrough that made the situation even more complicated. An independent laboratory conducted a deeper genomic analysis, focusing not on the full DNA sequence, but on specific segments related to cellular repair and stress response. These are the parts of human biology that activate under extreme conditions: injury, environmental stress, survival threats. In modern humans, these systems shut down after death. Completely. But in this case, traces of those systems appeared to remain structurally intact. Not active in the sense of sustaining life, but not fully degraded either. As if the shutdown process had been interrupted. Or paused. This is where the narrative began to shift. Because now, the question was no longer just scientific. It was conceptual. What does it actually mean for something to be dead? We tend to think of death as a moment. A clear boundary. Before and after. But biologically, death is a process. A gradual shutdown of systems. Cells die at different rates. DNA degrades over time. There is no single instant where everything stops. So what if, in rare cases, that process does not complete the way we expect? What if something interferes? Imagine standing in that laboratory, looking at a body that should have turned to dust thousands of years ago, and realizing that, at a microscopic level, parts of it never fully crossed the boundary we call death. Not alive. But not entirely gone. I try to understand what that would feel like, not as a scientist, but as a person. To look at something ancient and realize it is not just a remnant of the past, but a question that has not yet finished asking itself. It changes how you see time. It changes how you define endings. And then, just when it seemed like the mystery could not deepen further, another detail emerged. The burial itself. Closer analysis of the chamber revealed something subtle but intentional. The materials surrounding the body were not random. They formed a pattern, a deliberate arrangement that may have influenced the internal environment of the tomb. Chemical traces suggested the presence of compounds known to slow biological degradation. Not perfectly. Not completely. But enough to matter. It raised a possibility that is difficult to ignore. What if the people who placed this body here were not trying to preserve it as a memory? What if they were trying to preserve it as something unfinished? Not alive in the way we understand. But not meant to disappear either. And if that is true, then this is no longer just a scientific anomaly. It is a message. A signal from a time when the boundary between life and death may not have been seen as fixed, but as something that could be influenced, delayed, or even controlled. The DNA did not give a final answer. It gave a contradiction. A body that should be silent, still capable of response. A life that should be over, leaving behind systems that never fully shut down. And maybe that is the most unsettling part of all. Because it suggests that death is not as absolute as we believe. That under certain conditions, with the right biology, the right environment, the right intention, something can remain. Not alive. Not conscious. But not entirely gone. And if that is possible. If even one body can exist in that state, then the question is no longer about the past. It is about the limits of what we think is possible. Because somewhere, in a sealed chamber beneath the earth, a body waited for 3,000 years, and when we finally looked inside, it wasn't just history we found. It was a boundary that may never have been as solid as we thought. It was a boundary that may never have been as solid as we thought. And that should have been the end of the story. But it wasn't. Because after the initial reports were published, something unexpected happened. The data didn't remain contained within a single lab. It began to move quietly between research groups, universities, and independent scientists. And they all started asking the same question: what exactly is still active, and why? In the second phase of testing, scientists focused on one specific anomaly, the strange cellular response. Tiny samples of tissue were placed in a carefully controlled environment designed to simulate basic biological conditions. Nothing extreme. No artificial stimulation beyond what a normal system might encounter. In almost every known case, ancient tissue remains completely inert under such conditions. There is no reaction. No change. Just silence. But this time, there wasn't silence. At a microscopic level, the tissue showed fluctuation. Not life. Not revival. But not complete stillness either. Certain molecular signals appeared to react to the surrounding environment, as if some internal mechanism had not fully shut down. The activity was weak, inconsistent, almost fading, but it was measurable. And for scientists, that was enough to confirm that something here did not follow the known rules. At this point, a new theory began to take shape, what some researchers called delayed biological shutdown. The idea is simple, but unsettling. Death is not a single moment. It is a process. Systems shut down in stages. Cells break down over time. But what if, under extremely rare conditions, that process is interrupted? What if parts of the body never fully complete the transition into total inactivity? If that were true, it would mean this mummy is not just preserved, it is suspended at a biological threshold. But even this theory had limits. Because nothing in modern science suggests that human tissue can maintain this kind of partial responsiveness for thousands of years. Not without energy. Not without repair. Not without something sustaining it. And that leads to the most difficult question of all. What if the explanation is not just environmental, but intentional? Further analysis of the burial chamber revealed something subtle but deeply significant. The materials surrounding the body were not randomly placed. They formed a deliberate structure. Chemical traces indicated compounds that may have slowed cellular degradation in a very specific way, not stopping death, but interfering with how it completes itself. Almost like creating conditions where the body doesn't decay normally, and doesn't fully shut down either. Which raises a possibility that is hard to ignore. What if the people who prepared this body understood something we don't? Not in a mystical sense, but in a biological one. What if they discovered a way to place the human body into a state where it lingers between processes, no longer alive, but not entirely gone? A controlled pause, rather than an ending. Imagine standing in that chamber thousands of years ago, believing that death was not final, just incomplete. That with the right method, the right environment, the right preparation, you could hold on to something beyond its natural limit. And now imagine discovering that they might have been right. Even if only partially. Because this mummy does not prove immortality. It does not prove life after death. But it does challenge something we rarely question, the certainty of the boundary itself. If even one body can exist in this state, then death is not as absolute as we assume. It becomes a spectrum. A process that, under certain conditions, might be slowed, altered, or left unfinished. And maybe that is why this discovery matters. Not because it gives us answers, but because it removes certainty. Because somewhere in the past, long before modern science, someone may have tried to change the way death works. And for reasons we still don't understand, they may not have completely failed. If you found this story intriguing, make sure to like, subscribe, and share, because some mysteries aren't just about the past, they're about the limits of what we believe is possible.